everyone, I am Shwelidash welcoming you to the Medical and Pharmacy Crash Course Division in the Knowledge Club online. In the previous episode, we learned about various types of neurotransmitters. Today, I will discuss the various types of neurotransmitter receptors. By the end of this section, you will be able to know about the multitude of neurotransmitters used in the nerve system provides it with a specific and flexible interneuronal communication system. These characteristics are even further enhanced by the variety of receptors for each neurotransmitters. Stay tuned up to end because if you skip the lecture, you can miss a valuable points in between lecture for your exam. So stay tuned up to end. I am going to start today's episode. Let's start. Receptors for a particular neurotransmitter were traditionally distinguished primarily by pharmacological differences in their sensitivity to particular agonists and antagonists. For example, acetylcholine receptors were split into muscarinic and nicotinic classes, depending on whether they bind muscarinic or nicotine. Similarly, glutamate receptors were split into three main groups according to their sensitivity to the agonist: NMDA, carinic acid, or AMPA. Though useful, this classification scheme has several limitations. Some receptors fail to be activated by agonists, and it fails to disclose all the various receptor subtypes for a particular transmitter. Over the past 15 years or so, molecular biological approaches have been used to identify and sequence the receptor genes for many of the known neurotransmitters. It is thought that we now have a relatively complete catalog of the genes for these receptors. What this work has revealed is that there is a tremendous diversity of actual and potential receptor subtypes that are or could be used by the nervous system. Moreover, knowledge of the gene sequences has enabled an understanding of the relationship of different receptor proteins to each other and to other important proteins. This knowledge, combined with the results of biochemical, crystallographics, and other types of studies, has led to a much deeper understanding of the structural and functional workings of receptor proteins. In particular, various receptors can be grouped into families based on the gene sequences, and numbers of each family share various structural and functional features. Neurotransmitter receptors are members of one of two large groups or families of proteins: ligand-gated ion channel, also known as ionotropic receptors, and G-protein coupled receptors, also referred to as metabotropic receptors. Almost all classic neurotransmitters and neuropeptides have at least one metabotropic type receptor. Many of the classic neurotransmitters also have at least one ionotropic receptor. Ionotropic receptors are protein complexes that both have an extracellular binding site for the transmitter and form an ion channel that is poured through the cell membrane. The receptor is made up of several protein subunits, usually 3 to 5, each of which typically has a series of membrane-spanning domains, some of which contribute to the wall of the ion channel. Bindings of the neurotransmitter alters usually increases the probability of the ion channel being in the open state and thus typically results in post-synaptic events that are rapid in both onset and decay, with a duration of several milliseconds. Ionotropic receptors underlie fast synaptic EPSPs and IPSPs as described earlier episode. The ionotropic receptors can be divided into several superfamilies. Members of the cis-loop superfamily have peptide subunits that have an N-terminal extracellular domain that contains a loop delimited by cysteine residues. This family includes the ionotropic receptors for acetylcholine, serotonin, Gaba, and glycine. In addition to their family-defining cysteine loop, these receptors share the following common features. There are pentamers, with each peptide subunit having 4 transmembrane domains. The neurotransmitter binds to the N-terminal domain, and the second transmembrane domains are thought to form the wall of the ion pore. Ionotropic glutamate and ATP receptors form 2 other ionotropic receptors superfamilies. The details of each are given in the latter corresponding section of episodes. Transient receptor potentials, the TRP channels, which are important for transduction of pain and thermal sensations, form yet another family. Metabotropic receptors are not ion channels. Instead, they are protein monomers that have an extracellular binding site for a particular transmitter and an intracellular site for binding a G protein. Binding of the receptor levels to activation of a G protein, which is the first step in a signal transduction cascade that alters the function of an ion channel in the postsynaptic membrane. In contrast to ionotropic receptors, metabotropic receptors mediate postsynaptic phenomena that have a slow onset and that may persist from hundreds of milliseconds to minutes. Because of the various biochemical cascades they initiate, they have great potential to cause changes in the neuron beyond just generating a postsynaptic potential. So I am starting with the acetylcholine receptors. Acetylcholine receptors were originally classified on the pharmacological basis, being sensitive to nicotine or muscarine, into 2 major groups. This grouping corresponds to grouping based on structural and molecular biological studies. Nicotinic receptors are members of the ionotropic cis-loop family, and muscarinic receptors are part of the metabotropic family of receptor proteins. The nicotinic receptors mediate synaptic transmission at the neuromuscular junction as described earlier episode. However, the nicotinic receptors are also present within the CNS. The nicotinic receptor contain a relatively non-selective cationic channel, so binding of acetylcholine produces an EPSP. Being members of this cis-loop family, acetylcholine receptors are pentamers conducted from a series of subunit types called alpha, beta, gamma, delta, epsilon, some of which contain multiple numbers. At the neurotransmitter muscular junction, the channel is constructed from 2 alpha, beta, delta, epsilon, whereas in the CNS, the composition is typically 3 alpha, 2 beta. Furthermore, the junctional receptor all use the alpha 1 subunit, whereas the centrally located receptors use one of the alpha subunits between alpha 2 and alpha 10. As noted, the differing subunits result in the receptors with differing pharmacological sensitivities and channel kinetics and selectivity. There are 5 known muscarinic subtypes of acetylcholine receptors, that is M1 to M5. All the metabotropic receptors, however, they are coupled to different G proteins and can thus have distinct effects on the cell. M1, M3 and M5 are coupled to pertussis toxin insensitive G proteins, whereas M2 and M4 are coupled to pertussis toxin sensitive G proteins. Each set of G proteins is coupled to different enzymes and second messenger pathways. Next type of receptors are the inhibitory amino acid receptors. Gaba and glycine, as noted, the most common inhibitory synapses in the CNS was either glycine or Gaba as their transmitter. Glycine-mediated inhibitory synapses are common in the spinal cord, whereas GABAergic synapses make up the majority of inhibitory synapses in the brain. Both glycine and Gaba have ionotropic receptors that are members of the cis-loop family, thus sharing a number of characteristics as already described in the previous episode. In addition, each of these receptors has a chloride ion channel which opens while the receptor portion is bound. Therefore, the probability of these channels opening and the average time a channel stays open are controlled by the concentration of the neurotransmitter for which the receptor is specific. Glycine receptors are pentamers and may be heteromers of alpha and beta subunits, that is 3:2 ratio of homomers. Interestingly, the molecular composition appears to be related to its cellular location, with heteromers located post-synaptically and homomers located extrasynaptically. The beta subunit seems to bind to an intracellular scaffold protein called gephyrin that appears to help localize receptors to the postsynaptic site. The alpha subunit contains the glycine binding site, and there are 4 genes coding for distinct alpha subunits and splice variants of each. Each variant results in a receptor having distinct conductance, kinetics, agonist and antagonist affinity, and modulatory sites. Intriguingly, subunit variants are differentially expressed during development and in different brain regions. Gaba has 2 separate inotropic receptors, that is GABA and GABAC, coded for by distinct sets of genes. Like glycine receptors, both control a chloride ion channel. GABA receptors are heteromers generated from even classes of subunits, 3 of which have multiple members. The most common configuration is the alpha 1, beta 2, gamma 2 in a 2:2:1 stoichiometry, which may account for 80% of the receptors. However, many other heteromers are found in the brain. As with glycine, different subunits confer distinct properties on the receptor. For example, GABA receptors are the targets of 2 major classes of drugs, the benzodiazepines and barbiturates. Benzodiazepines, that is the diazepam, are widely used anti-anxiety and relaxant drugs. Barbiturates are used as sedatives and anticonvulsants. Both classes of drugs bind to distinct sites on the alpha subunits of GABA receptors and enhance opening of the receptors chloride ion channels in the response to GABA. The sedative and anticonvulsant actions of benzodiazepines appears to be mediated by receptors with the alpha 1 subunit, whereas the anxiolytic effects reflect binding to receptors with the alpha 2 subunits. GABAC receptors are structurally similar to GABA receptors but have a distinct pharmacological profile, that is, they are not affected by benzodiazepines and are coded for by a separate set of genes, that is Rho1, Rho2 and Rho3. The GABA B receptor is a metabotropic receptor. Binding of GABA to this receptor activates a heterotrimeric GTP binding protein, which leads to activation of potassium ion channels and hence hyperpolarization of the postsynaptic cell, as well as inhibition of calcium ion channels when located presynaptically, and thus a reduction in release of transmitter. Next type of receptor is the excitatory amino acid receptor, that is glutamate. Glutamate has both inotropic and metabotropic receptors. Based on pharmacological properties and subunit composition, several distinct inotropic receptor subtypes are recognized, that is AMPA, kinase and NMDA. Overall, there are 18 known genes that code for glutamate subunits for the inotropic glutamate receptors. The genes are divided into several families: AMPA, kinase, NMDA and delta, that essentially correspond to the pharmacological subtypes of receptors. Each glutamate receptor is a tetramer. Thus, there is a certain correspondence between the genes and the receptor types that are formed. For example, AMPA receptors are formed from GluR1 to GluR4 subunits. Kinase receptor require either Ka1 or Ka2 and GluR5 or GluR7 subunits, and NMDA receptors all have NR1 subunits plus some combination of NR2 and NR3 subunits. As was mentioned for the other chapters, the receptor properties vary with subunit composition. Inotropic glutamate receptors are excitatory and contain a cationic selective channel. Thus, all the channels are permeable to sodium ion and potassium ion, but only a subset allow calcium ion to pass. AMPA and kainate receptors behave as classic ligand-gated channels as already discussed in the previous episode. On binding of glutamate to the receptor, the channel opens and allows current to flow, thereby generating an EPSP. NMDA channels are different. First, they require binding of both glutamate and glycine to open. Second, they display voltage sensitivity as a result of magnesium ion blockage of the channel. That is, at resting or more negative membrane potential, magnesium ion blocks the entrance to the channel so that even when glutamate and glycine are bound, no current flow through the channel. However, if the cell is depolarized either experimentally by injection of current through an electrode or by other EPSPs, the magnesium ion block is relieved and current can flow through the channel. A further interesting feature of NMDA channels is that they are generally permeable to calcium ion, which can act as a second messenger. The combination of voltage sensitivity and calcium ion permeability of the NMDA channels has led to hypotheses concerning their role in learning and memory related functions. 8 genes coding for metabotropic glutamate receptors have been identified and classified into 3 groups. Group 1 receptors are found postsynaptically, whereas groups 2 and 3 are found presynaptically. These receptors generate slow EPSPs, but probably at least as important, they trigger second messenger cascades. Third type of receptors is the purine, the ATP receptor. Purines have 2 receptor families as inotropic P2X and a metabotropic P2Y family. There are 7 identified P2X subunit types that form channels, and they represent their own superfamily of ligand-gated channels. Each subunit has only 2 transmembrane domains, with the loop between these 2 domains located extracellularly and containing the ATP binding site. The receptors are heterotrimers or homotrimers or hexamers. In general, these receptors form a cationic channel that is permeable to sodium ion, potassium ion, and calcium ion. The distribution of subunits in the brain varies significantly, with some subunits having a widespread distribution, P2X2, and other being quite limited. The P2X3 is present mostly on cells involved in pain-related pathways. Metabotropic purine receptors are coded for by 10 genes, but only 6 are expressed in the human CNS. They have the typical features of G-protein coupled receptors and are known to activate potassium current and modulate both NMDA and voltage-gated calcium current. An interesting localization distinction between P2X and P2Y receptors is that although both are present in neurons, the latter dominates on astrocytes. Finally, the addition to the P2X and P2Y receptors which respond to ATP. There are adenosine receptors that respond to the adenosine that is released after enzymatic breakdown of ATP. These receptors are located presynaptically and act to inhibit synaptic transmission by inhibiting influx of calcium ion. Fourth type of receptor is the biogenic amine receptors, that is serotonin, dopamine, noradrenaline, adrenaline, histamine. With the exception of one class of serotonin receptor, 5-HT3, that are part of the cis-loop inotropic family. Receptors for the various biogenic amines are all metabotropic type receptors. Thus, these neurotransmitters tend to act on relatively long time scales by generating slow synaptic potentials and by initiating second messenger cascades. Agonists and blockers of many of these receptors are important clinical tools for treating various neurological and psychiatric disorders. The role of different dopamine receptors in basal ganglia disorders will be covered in the motor system. Fifth type of receptors, that is neuropeptide receptors. As is the case with the biogenic amines, receptors for the various peptides are essentially all of the metabotropic type and are coupled to G protein that mediate effects via second messenger cascades. It is worth mentioning again that studies consistently show a mismatch between the locations of terminals containing a particular peptide and the receptors for it. Thus, these receptors are often activated by neurotransmitter diffusing through the extracellular space rather than by synapses. This implies that these receptors will experience much lower concentrations of agonists, and indeed they are very sensitive to their agonists. The last type of receptors is the gas neurotransmitter receptors. Unlike the other neurotransmitters that were covered, NO and CO do not bind to receptors. One way they do affect cell activity is to activate enzymes involved in second messenger cascade such as guanylyl cyclase. In addition, NO has been shown to modify the activity of other proteins such as NMDA receptors and the sodium-potassium ATPase pump by nitrosylating them. I am finishing today's episode up to this. Revise it thoroughly. I am very sure you will require less time to remember all these structures and functions. Write your opinion to me at the comment box how much you enjoyed and time take to remember the nervous tissue anatomy physiology after viewing today's lecture. This episode we will discuss anatomy of the central system in the next episode. If you like this video, please like, share and comment freely. Don't forget to subscribe our channel. If you already subscribed, then thanks a lot. After subscribing our channel, please click on the bell button for getting the regular updates. I am coming with another video very shortly. Till then, goodbye.